Good day, this is Professor Will, CBMD PhD. Today is Saturday, May 29, 2021. It is 2:14 and 50 seconds a.m. Eastern Standard Time. And today's research, subatomic particles. So let's hop to it here. Algorithm to see inside materials with subatomic particles. Date May 17, 2021. Source, University of Kent. Researchers have developed an algorithm to train computers to analyze signals from subatomic particles embedded in advanced electronic materials. The particles, called muons, are produced in large particle accelerators and are implanted inside samples of materials in order to investigate their magnetic properties. Muons are uniquely useful as they couple magnetically to individual atoms inside the material and then emit a signal detectable by researchers to obtain information on that magnetism. This ability to examine magnetism on the atomic scale makes muon-based measurements one of the most powerful probes of magnetism in electronic materials, including quantum materials such as superconductors and other exotic forms of matter. As it is not possible to deduce what is going on in the material by simple examination of the signal, researchers normally compare their data to generic models. In contrast, the present team adapted a data science technique called principal component analysis, PCA, frequently employed in face recognition. The PCA technique involves a computer being fed many related but distinct images, and then running an algorithm identifying a small number of archetypal images that can be combined to reproduce with great accuracy any of the original images. An algorithm trained in this way can then go on to perform tasks such as recognizing whether a new image matches a previously seen one. Researchers adapted the PCA technique to analyze the signals sent out by muons embedded in complex materials, training the algorithm for a variety of quantum materials using experimental data obtained at the ISIS neutron and muon source of the STFC Rutherford Appleton Laboratory. The results showed the new technique is equally as proficient as a standard method at detecting phase transitions, and in some cases could detect transitions beyond the capabilities of standard analysis. Good day, this is Professor Will, CBMD PhD, of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.